The organ systems we have discussed thus far function continuously to maintain the well-being of the individual. The reproductive is inactive until puberty and functions to propagate the species. In this presentation, we will look at the primary and secondary sex organs, how gametes are produced, and the first early stages of pregnancy. The term primary sex organs is applied to the structures of both the male and female reproductive systems that produce the gametes and secrete hormones that support their function. At this point you may be asking yourself a couple of questions. Why do the testes lie outside the pelvis whereas the ovaries of females reside securely within the pelvic cavity? Also, why does the vas deferens loop over the ureters to go behind the bladder in order to pick up secretions from the seminal vesicles? The answer to the first question is that sperm production takes place at 3 degrees centigrade below body temperature. Why this is so, God only knows and He ain't telling. The answer to the second question is a little easier to understand. By week 7 or 8, a couple of mesodermal masses of tissue within the abdominal cavity called the urogenital ridges have begun developing into the kidneys, adrenal glands and the testes. The testes are held to the abdominal wall by the genitoinguinal ligament. As fetal growth continues, the trunk elongates, but the genitoinguinal ligament does not grow as fast. This causes the testes to descend from the abdomen so that by the 5th month of gestation, the testes are even with the inguinal ring. By this time, the anterior abdominal wall has evaginated to form the scrotum. The testes continue their descent through the inguinal canal so that by the 8th month they lie in the scrotum. Because the testes develop retroperitoneally, they carry with them all the layers of the abdominal wall which end up in the scrotum and eventually become the coverings or tunics of the testes and spermatic cord. The testes are suspended in the scrotal sac which is an evagination of the body wall. The layers of the scrotum from outside to inside consist of skin containing a thin cutaneous muscle called the dardos, superficial scrotal fascia and another muscle called the cremaster. Both the dardos and cremaster muscles function to help regulate the temperature within the scrotum to support optimal sperm production. The outer capsule of the testis consists of some shiny, dense CT called the tunica albuginea. Each testis is divided by septi into about 250 lobules. Each lobule contains between 1 to 4 coiled tubules called the seminiferous tubules which are the site of sperm production of spermatogenesis. The picture on the left above shows a photomicrograph of a seminiferous tubule. Each tubule is surrounded by a connective tissue capsule. Inside the tubule there is a complex epithelium consisting mainly of spermatocytes in various stages of development, and the nuclei of a few tall, irregular cells called sustentacular cells. The picture on the right is a diagram of a similar cross-section through a seminiferous tubule that shows the cells and supportive structures more clearly. Note that the capillaries are on the outside of the seminiferous tubule. There are also some large cells called interstitial cells, Leydig cells residing nearby but outside the seminiferous tubule. The interstitial cells are the source of testosterone which directs the development of sperm and is responsible for maintaining the accessory glands, as well as male secondary sexual characteristics. Let's take a closer look at an enlarged portion of the seminiferous tubule epithelium. The first cells that are encountered sitting on the connective tissue capsule and diploid or 2 in spermatogonia. The spermatogonia are the stem cells for all the sperm a male will make throughout his lifetime. As we move toward the lumen of the tubule, we see spermatocytes in various stages of meiotic division and maturation. The lavender-colored sustentacular cells support the developing gametes both physically and physiologically. They also form a blood-testis barrier which protects the spermatocytes from attack by immune cells and maintains the appropriate chemical environment for optimal sperm production. We will see later that the sustentacular cells also secrete hormones that help to regulate testosterone levels within the seminiferous tubule. Spermatogenesis is the process of making mature haploid in sperm for the diploid spermatogonia. The process is divided into 3 steps. Spermatocytogenesis involves mitotic division of the spermatogonia. Recall that the spermatogonia are the source of all the sperm a male will make. Therefore, the spermatogonia must make identical copies of themselves via mitosis. When a spermatogonium divides, one of the 2 daughter cells stays behind to remain as a spermatogonium and the other begins its development toward becoming a mature sperm. The daughter spermatogonium will then undergo 2 successive cell divisions resulting in the formation of 4 haploid spermatids from a single spermatogonium. This process of reduction division is called meiosis. During meiosis the number of chromosomes is reduced to half, from 46 to 23. Each diploid cell of our body has 46 chromosomes or 23 pairs, one set of the pair from the mother and one set from the father. Fertilization and formation of a zygote restores the diploid number. If there were no meiosis, then each successive generation of offspring would double their chromosome number. 46 plus 46 equals 92, 92 plus 92 equals 184, etc. Variations in the chromosome number of a species leads to either death of the embryo or severe genetic disorders. The next step of spermatogenesis is called spermiogenesis. This process involves an extreme makeover and reorganization of the spermatid cytoplasm to form a mature, mobile sperm cell. This picture illustrates the dramatic changes that take place in structure of the spermatids during the process of spermiogenesis. The Golgi apparatus produces hydrolytic enzymes necessary to break down the oocyte's membrane and forms the acrosomal vesicle. Meanwhile, cytoplasm is being shed and reorganized. The nucleus condenses and the acrosomal cap moves to the region that will become the head of the sperm. The mitochondria reorganize and form a spiral around the base of the tail. The tail is a flagellum and the mitochondria supply the energy that propels the tail. Here's a closer look. This picture shows the end result of spermiogenesis. Note the acrosomal cap will be the part of the sperm that contacts the oocyte first. The nucleus is greatly condensed and there is very little cytoplasm. The mitochondria form a spiral around the midpiece, which will power the tail, which consists of a flagellum. There is a lot of overlap between the endocrine system and almost every organ of the body, and the reproductive system is no exception. We will cover the endocrine system in physiology, but there exists crosstalk between the hypothalamus, pituitary gland and the testes. This is called the brain-testicular axis, which takes about 3 years to get regulated. The function of the brain-testicular axis is to balance male hormones to ensure that enough sperm are being produced, and to regulate secretions of the glands and maintenance of male secondary sex characteristics. When the sperm count falls below 20 million/ml of ejaculate, the male is essentially sterile. Conversely, overly high levels of testosterone are harmful for normal spermatogenesis and other functions. Here's the short version of how optimal levels of testosterone are maintained both in the bloodstream and within the seminiferous tubule. The cells within the testes that are involved are the interstitial cells and the stentacular cells. Under command of the hypothalamus pituitary gland, the interstitial cells secrete testosterone. The testosterone enters the bloodstream via the capillaries located outside the seminiferous tubules. This testosterone maintains target tissues associated with being male. Testosterone is also needed inside the seminiferous tubule to support sperm production, however the cystentacular cells form a blood-testis barrier. Therefore, the cystentacular cells produce and secrete androgen-binding protein, which combines with testosterone and pulls it into the seminiferous tubule. When testosterone levels within the seminiferous tubules get too high, the cystentacular cells secrete a hormone called inhibin, which stops the release of hormones from the hypothalamus pituitary complex so that there is not too much testosterone within the testes. When blood levels of testosterone get too high, it feeds back to the hypothalamus pituitary gland, which slow down their release of hormones that cause its formation. From the lumen of the seminiferous tubules, the sperm move into some short straight tubules and then to a meshwork of tubules called the rete testis rete, like reticulum means net-like. From there the sperm move into about 12 to 15 efferent ductules, which coil up to form the head of the epididymis. The epididymis is a comma-shaped structure comprised of a head, body and tail that actually consists of 20 feet of a single coiled tube that is only 1 millimeter in diameter. The epididymis straightens out, forming the ductus deferens, which travels in the spermatic cord and enters the pelvic cavity after passing through the inguinal canal. The vas deferens and several other structures pass together from the scrotum through the inguinal canal. In addition to the vas deferens, the testicular artery, genitofemoral nerve, autonomic nerves and a complicated system of veins called the pampiniform plexus are wrapped together and covered by the cremaster muscle and the connective tissue of the abdominal cavity. Both the cremaster muscle, and the dartos muscle of the scrotum are important for maintaining the optimal temperature for sperm production. The cremaster is actually part of the internal oblique abdominal muscle that was pulled along when the testes descended. Recall that sperm production takes place at 3 degrees centigrade below body temperature. When the temperature in the scrotum gets too high, the cremaster muscle relaxes and the testes drop down further into the scrotal sac away from the body. When it is very cold, the cremaster contracts, pulling the testes closer to the body to help them stay warm. The dartos relaxes when it is hot, and the testes move outward from the body. However, when it's cold, the dartos contracts, wrinkling the skin of the scrotum, which pulls the testes closer to the body for warmth. Ejaculation is mediated by the sympathetic division and involves two steps. First, there is a sudden contraction of all the smooth muscle that makes up the walls of all the ducts and glands. This delivers the semen to the urethra. Semen is then forcefully expelled from the urethra by the contraction of the bulbospongiosus and ischiocavernosus muscles, both are skeletal muscles. Prostate cancer is the leading cause of cancer death in U.S. men. In the past, acid phosphatase levels in the blood were used to help diagnose the presence of malignant tumors, but many tissues in the body can produce acid phosphatase. The discovery of prostate-specific antigen led to the development of a more specific test. PSA is a proteolytic enzyme recently discovered that is specific to the prostate gland. PSA normally functions to liquefy semen. Recall that semen congeals within 5 minutes following ejaculation because of the semenogelin produced in the seminal vesicles. PSA and the other proteolytic enzymes help to solubilize it so that the sperm can move through the cervix and uterus. PSA can be detected in the blood. Levels of PSA increase with an increasing size of the prostate. Prostatic enlargement may be benign. There are mucus glands around where the urethra passes through the prostate, and in older men these can become enlarged and pinch off the urethra, making urination difficult. However, prostatic enlargement may be due to cancer. Although the PSA test is not perfect, it can detect prostate cancer before the tumor is large enough to cause urinary problems and while it is too small to be detected with a digital rectal exam (DRE).